So my main project is carbon nanoparticles and their effect on plant-to-plant communication. So this is a joint project between the Biomedical Engineering Department under Dr. Prabir Patra and also Dr. Kathleen Engelman in the Bio Department here at UB. So I'm also working with another grad student, Ashwini, who couldn't make it here today, but we're working together on this project. So who has seen this movie? Just by raise of hands. Okay. Not as many as I would like, but it's a great movie. Jurassic Park, really recommend you guys watch it. Very, one of my favorites. But it's actually based on a book that was written in the 1980s by Michael Crichton, who was a very famous techno thriller. So he writes a lot of really heavily scientific work. And there's a quote from the book that I wanted to include here. People were so naive about plants. They just chose plants for appearance because they would choose a picture for the wall. It never actually occurred to them that plants were actually living things, visually performing all the living functions of respiration, ingestion, excretion, reproduction, and defense, which is very true. And if you guys look outside right now, you will see a plant as a backdrop. But in reality, plants are very much living things just like you and I are. And as such, they can be good for us, they can be bad for us, they compete with each other, they fight with each other, they mate with each other. They're very much part of the animal kingdom just like you and I are. And the reason why I wanted to include that quote here is because plants communicate. They talk, but not in ways that you and I talk. And their way of sensing the environment is very unique. They, like us, have their own senses, their own ability to transduce signals that they see in the environment, their own ability to perceive what's going on. And they're also transduced via inter- and intercellular signals, just like we do. So this experiment right here is an example of plants being used as a biosensor. So plants are actually very sensitive to their environment. I mean, right now, if this is your first time in America during the fall, you may have noticed the leaves changed color very quickly. That's again, the plants reacting to the dropping temperature of our environment. And that's an example of how plants can kind of adapt themselves rapidly, much more rapidly than humans or most animals can, to changes in the surrounding environment. And the crazy thing is these signals are not just limited to one plant. So within this experiment, they wired up this plant to some electrodes, and they basically measured electrical impulses down the phloem. The phloem is basically part of the vascular system of a plant, and it kind of functions as a nerve bed, just like we have. And they were basically measuring the plant's response to light, using it to detect changes in light like a biosensor. And it's cool because this experiment only was done within one single plant unit. When in reality, plants can also talk to each other. This is called wired and wireless communication. So this paradigm is very important to consider. Talk because over the air, over long distances. I'm talking about kilometers here, being able to communicate with plants that are not even within the same vicinity. This term of wireless communication. So my specific project talks about wired communication via mycorrhizae, which I will go into now. And this is just an example of some plants communicating. I like this picture a lot because it shows plants reacting to different things in their environment. So this can be threats like pathogens, pests, bugs, insects. It can be things that help the plant, sunlight, water, nutrients, and also different ways the plants talk, right? So these are kind of direct communications. So this right here is mycorrhiza, which is what I'm going to be focusing on getting into in a little bit. But also indirect, looking at daughter, looking at how plants release particulate that can communicate to plants down the line, being able to communicate across vast distances. So it's pretty cool that we're able to do this over such a long, both short and long. So what is a mycorrhizal connection? So mycorrhiza are fungal roots. So you may have heard when you guys are taking your biology classes, you may have heard of mutualism, parasitism, and symbiosis, right? Different ways. So mycorrhiza has a symbiotic relationship with many species of plants. I'm going to go show you guys an actual picture of this in a while. And once you see the picture, you'll absolutely recognize what they look like because you've definitely seen them before in your life. And they're a fungal root. So, 1 gram of soil in most parts of the world contains 10 to 100 molar of this fungi. And basically what this fungus does is it creates connections, kind of like an information highway between two sets of roots, between two neighboring plants. And the plants can take advantage of this highway, so to speak, to spread information from one plant to the other. The mycorrhiza will be able to gain access to nutrients, be able to kind of have a place to host and live, and the plant gets access to communication. An example of this is Tagetes tenuifolia. It can secrete thiophene, which is I think it's a hydrophobic molecule, more than 12 centimeters via these connections. And that's a pretty big distance when you're thinking about how thin and small these mycorrhizal connections are. And these signals can have effects on neighboring plants. So I pulled this image from a paper. where they basically exposed potato plants to a pathogen. And they had these plants, it's actually a very similar from the plant that was infected via these mycorrhizal roots. So here, this is just like a gene hot map of genes that were upregulated. So JA/ET, this group was more for defense against this pathogen. And if you look down here, so they have a whole different bunch of parameters, but this is the donor, that's what D is, and then this is the recipient, that's what R is. M means that they had a mycorrhizal connection, so M+. They also cut some of the plants, so if they were cut, there was no connection, so you wouldn't expect to see any sort of DNA. And as you can see, these defense enzymes, or these defense genes were so much regulated compared to everything else because they just were having access to the plants that were infected. It gives kind of, proof that these plants are talking to each other. And it's pretty cool that that actually happened. So these are pictures of mycorrhizal connections. And again, if you guys have, who's planted a plant before? Bought a flower from a nursery, planted it in the ground. You've definitely seen fibers like this. Most, even lawn experts and garden care experts will recommend that you allow these fungi to grow within your garden because they're actually good for your plants. It helps them grow and be healthy. So yeah, it's like this little white fibers. Again, it's like the diameter of a hair, very thin. So it's good actually for the plants to have these.
Maybe it would be a very good idea. Do you know anybody has done this or not? The structural imaging of these mycorrhizal connections, how small they are, they should be very small.
Mm-hmm. Yeah, I should have included maybe the towns.
No, that's nice.
It's very cool because you guys have definitely seen some connections to travel from one plant to the next. And there's many reasons why we're curious about this. I actually found a lot of papers. So I have a patho background, as many of you know. I don't really know a lot about plants, but as I was doing a lot of research, I noticed that there's a lot of applications of carbon nanoparticles within agriculture. For example, using nanoparticles to improve plant growth, resistance against disease and pests. That's what this paper is here for. They're using carbon nanotubes polymerized with polymer chains that allows it to catch plasmid DNA, and using that as a delivery mechanism for plasmid DNA barrier to introduce foreign DNA into these plants. But they were able to do so and get into the mitochondria and actually do recombination within the mitochondrial DNA. Same with this. They used, again, a single wall carbon nanotube and plasmid DNA complex together, able to get to the stomata, which are little holes in the cell wall, to get into the chloroplast and actually change the DNA of that.
Does this marketer's work?
Huh?
Does this marketer's work?
I don't think so.
Not this one.
Yeah. Another thing that's actually growing field is seeing how nanoparticle pollutants can affect plant growth. Industrial, chemical industrial product, there's a lot of a higher chance that it'll actually end up in our atmosphere. You guys have probably seen about microplastics and how a lot of it's ending up in fish and seafood and in our own bodies. The same thing could happen to plants. So we need to find out how nanoparticles can affect the growth of plants. Is it toxic? Will it end up in our own food supply? Because obviously plants are such a huge part of our daily ingestion. So the base question is we want to see if carbon nanoparticles can use the same mycorrhizal communication networks that natural bacteria and enzymes do. So this is just some studies of nanoparticles in plants, just kind of building off of what we've talked about before. So this is principal component analysis of Thale-Crest genomic response, and they're looking at water, single wall nanotubes. This is just like the control, that means there was nothing applied at all, and also single wall nanotubes conjugated with plasmid DNA. And this experiment was interesting because they're not just looking at the effects of the DNA itself, but also the level of harm and genomic recombination that can occur when you apply different types of nanoparticles. So as you can see, water and single wall nanotubes had a very similar response, indicating that these carbon nanoparticles were well uptook by the plants. They didn't cause any undue chemical recombination or genetic recombination. And then this was also interesting because it looked at geometric shapes of nanoparticles. They used the square, a cuboid, a rectangular shape, a prism, and circles, and seeing how these gold nanoparticles can diffuse through watermelon plant systems, all the way from the leaves down to the roots. So this is my experimental proposal. Can nanoparticles, specifically graphene quantum dots, diffuse through mycorrhiza between two neighboring plants? So we're using graphene quantum dots because, as you know, they fluoresce naturally, so we were able to detect the fluorescence in the neighboring plant if we were able to kind of get that. And also, how do differently shaped nanoparticles diffuse the mitochondrial connections? Maybe looking not just at quantum dots, but also maybe some single wall nanotubes, multi-wall nanotubes, different shapes to see how they also diffuse. And this is just a TEMS image of quantum dots. So this is our experimental setup. So we're using spider plants. You guys have definitely seen spider plants before. They're like kind of long, friendly, very tropical looking plants. We have them in 2 neighboring planter boxes. They are in the lab over in Dana. And in between, there's this little square box here. That box is intended for the root systems of both of these plants to intermingle. So basically the intention is microzoal MC connections can basically form in this little box right here, allowing these plants to communicate with each other. That's the goal. So I've created this very hideous box contraption. The idea here is we will make a solution of DI water and quantum dots, spray it on one plant, use this contraption to prevent spray on the neighboring plant, and then maybe after 24 hours, 48 hours, different time scales, assess fluorescent in the neighboring plant. See if any of the quantum dots are able to diffuse down through the root system, through this little box here where the MC is, and up into the neighboring plant. And so basically we're going to be using like a spray bottle, like an industrial spray bottle that you use to spray cleaner, so nothing really special. One problem with that is we can't be certain that the concentration of nanoparticle we're giving to the plants is standard across all the different trials. So one possible kind of really backwards way of my thought for kind of saying how much is being sprayed is by spraying the spray bottle we'll be using multiple times onto maybe like a scale and determining liquid volume being injected and calculating density just to see how much exactly is being injected within one spray of the trigger. But again, if you guys have any thoughts on that, that'd be appreciated. And also the small replicant pool, only enough plants for about 6 separate trials. So that's a little more difficult to solve. Dr. Engelman and I are trying to consider more ways maybe to see if we can expand the pool, but again, these plants take so long to grow. The ones that are in the lab
Just make sure that we are very nicely dispersed in water.
Yeah.
Normally, these quantum dots, they'll be settling down beneath if there's no functional groups in this. So if they don't disperse, then add some SDBAs or SDS specified.
Yeah.
I think it will be very fabulous experiments. We can prove indeed it is going to the neighboring plant. Then our communication channel is proven, right?
Yeah.
So that'll be cool. And then, as you said, shape changed particles, quantum dots. They are